Okay, so what I was going to talk about last time was some of the basic filters and the filtering considerations. That didn't work. And then so I'm going to start with that and then talk about this session, something else. What's some considerations for different filtering questions, and then talk about tomorrow about what do you need to think about when you're setting up and calling SNPs. So yes, so what I wanted to point out here, so I have my hybridization analysis, and on each of those, so that I did structure, and I ran an analysis, or did a set of filtering, and then a refiltering for new hybrids. For the populations, I had one set of filtering for my population structure analyses, and then a different set of filtering for my diversity analyses, and then a whole another set of types of filters for my phylogenetic analyses. So I'm first going to just start with talking about some of these filters, and it's kind of, what is it? So filtering by loci. So that's removing a particular locus from your data set. And it's basically because they didn't work to the extent you wanted across all your individuals, and they can add a lot of noise to a lot of data sets. And data sets can be really variable, or analyses can be really variable on how sensitive they are to noise. Like Arthur was saying, PCOAs, which I hate and don't really want anyone to ever use, are really sensitive to this. Things like phylogenetic analyses and structure are kind of, they go really, really slow. But things like we're going to do in a minute, like Tajima's D, actually, if the code is written, it's not that sensitive to it at all. So you need to have a trade-off between the strength of the filter and the number of loci you need, and different metrics matter. So strong filters actually disproportionately remove heterozygous sites. Okay, and many population structure methods also work better with none. So this filter can vary a fair bit based on what your actual analysis endpoint is going to be. Another one is filtering by individuals. So this is, of course, removing an individual who didn't work very well. And it's important because it deletes the key thing you actually care about, which is the individuals. But it's also important because it removes individuals that may be a bit misleading because they didn't work well. But I will say this is the filtering step you will do that will cost you the most amount of money, right? Individuals are what you spend money on. So I tend to use this judiciously, and I often do this as a later step, at which I'm going to show you in a little bit. So filtering for reproducibility is again, we've talked about that. So individuals are duplicated in your data set. So you actually sequence the same individual independently more than once. And then across those individuals, you line up the same gene and ask, did you get the same answer? Did you get the same alleles for the same individual at that gene? All right, and so if you did, great. Right? And so that's important because there's lots of reasons you might not get that. And that could be things like you have unknown gene duplicates in your genome, and they both amplified, but they actually both have mutations. And so that's actually not the same information. So reproducibility tells you like this. So diversity arrays does that automatically for you as part of the service. If you're using AGRF, you have to ask for it and pay for it, but it is pretty fundamental. So I just sent off a bunch of sunset frogs. If you don't know what a sunset frog is, you should. They're amazing. If you want to see Australia's purple and orange frog, that's what it is. And so I could send off 94, and I had 66 individuals sent off, and all the rest of that was just to duplicate the genome. Especially since this frog has a big genome, because frogs are jerks in that way, and so the probability of like duplications is really high, so we want to be able to see if that's a problem. Another one is read depth. So that is the mean number of reads at a particular locus. So if you have a gene and you're doing all this sequencing, what's the stack of reads that inform that locus? And so that tells you how confident you can be in your base calls. So a low read depth is actually quite difficult if you're interested in heterozygosity, so you need to think about it like, okay, we have a read depth. And if you're using something like stacks, which I'll talk a little bit about tomorrow, so if you're gonna do your own SNP calling, you've got a gene and you have 2 alleles. And at that locus, you get a read depth of 10. So on average, you get 5 reads per allele. But what if your read depth is only 4 and you get 3 reads that say it's one allele and 1 read that says it's the other? So what happens is you kind of lose confidence. How do you know at a read depth of 4 that that's actually a heterozygous site and not a sequencing error? So read depth is important if your questions are about diversity, and they're less important about structural kinds of questions. And very high read depth can often suggest paralogs. So I was showing this. This is one of my data sets. So this is, the bars is how many of your SNPs had a certain read depth. Yeah? Oh yes, point on this. Sorry, the university lecturer comes out. Okay, so this is how many SNPs you have. So this is like 15, 20,000 SNPs that have a read depth from here to about 25, but then some of them go up to 3,000. So I don't really believe that those little gene fragments with 3,000 reads are all one gene, so I want to get rid of them. And so then you basically cut them out. And you can see this doesn't go down to 0 because you also want ones you're confident in, so I've cut that from about 15 to 100. So I only kept SNPs where the read depth for that was between 15 reads for a locus and 100. To get rid of those potential gene duplicates while also being confident in heterozygosity. So anyone, questions? Okay. The next is filtering out minor alleles. I say this in the next bit, but you read the literature. We've just did a minor allele filter. This is my I randomly typed something motion. We got rid of minor alleles that are less than 0.05. So this is called a site frequency spectrum. So this is your minor allele frequency and your number of SNPs. So you've got lots of SNPs that are below 0.05 in these 3 populations from this random paper that I found on the internet, which is down here if you want to look. So getting rid of singletons is really important, or important to understand. So this is where theory comes into it. Is the metric that you are going to calculate dependent on rare alleles? Because a lot of them are. So firstly, 0.05, if you have a ton of individuals, like if you had 300 individuals or 3 plates, and you delete SNPs that have a 0.05 minor allele frequency, you're requiring at least 30 copies of that allele before you keep it. And that might be a really high number. Are you really not confident about that allele in the population if there's 30 copies of it? So I think it's important to evaluate these. It can add a lot of noise, and I will go into that, for analyses that don't require rare alleles, but they're vital if you need them. Yes? So how does this relate to sample size of your population? It is 0.05 as a proportion. So if you are writing this for 0.05, it would just be it would require that, let's say so if you had 300 diploid individuals, you would have 600 copies of the allele in your 300 individuals, and 0.05 is 30 copies.
I've been recommended to look also not just at your total number of individuals. So if you have like 300 individuals, but they are subsetted into 10 locations, your 10 original populations, let's say, and each population would have, I don't know, 30.
Yeah.
So do you choose your math filter depending also on the number of individuals you have in each population or not? Because I know you can filter math by population or by like overall.
Yeah, so it's a good question. So let's say you have 300 individuals, but you have what you think are 12 separate populations. If you actually applied a 0.05 to that, you would require an allele to be present in at least 2 populations by applying a 0.05 filter, right? So if you want to be able to isolate alleles that are only within an individual population, your minor allele frequency filter can't be higher than the number of individuals in the population. Does that make sense? Yeah, so if you have a whole bunch of populations and each population is only 4% of your total individuals, then if you put a 0.05 minor allele frequency, it means that you are forcing the ones you keep to share alleles, right? Because the number of alleles that you keep is higher than the number of individuals in your populations. So you actually reduce your ability to probably distinguish. I don't know if anyone else wants to comment on that. You're good at graphs. Feel free to join. This is your session too. Yeah. Yes.
I'm not sure how people online are going to see this, but I picture this in this way. Oh, this doesn't work.
Oh yeah. The classic whiteboard marker. And you put it back, just like everyone else does. Describing it online. Oh, this is starting to be like art. All right, so
that's it. So if you have three sites, 3 populations, the one allele occurs only here. All right. In this case, I have 1, 2, 3, 4, 5 individuals with 1 copy of the allele you're after. All right. If you force the minimum allele frequencies to a value that's higher than this, all those individuals that carry the allele are not considered because you don't have enough copies in your data sets, right? So you're only retaining the alleles that are shared across the sites because that way you can get to the minimum number of instances of the same allele you're looking for.
So you're saying that you're filtering across population,
Depending on the threshold you use, that might happen. That's what Renee was correctly pointing out.
okay.
Does it make it easier to follow?
Yeah. Rob, you had a...
Does it make it easier to follow that way? If you picture that in your mind that way? If you don't use allele frequencies but use allele counts, it's easier, right? If you say, I want to have that allele present at least 6 times in your data sets, because of this purple allele you only got 5, you get rid of it. So those are gone. They're not there anymore. You don't see that difference between the sites anymore.
Yeah.
Make sense?
Please,
yeah.
Guys, use the microphone if you have a question. I know it's a pain, but online people can't hear that.
Yeah, and this comes back to something I'll touch on through this is you actually have to play with your numbers all the time. You have to do this multiple times to see how you're affecting your answers to try and figure out whether you're getting a biologically reasonable answer. So this is population genetics, but if it gives you a nuts balls answer, you've done something wrong. Yeah. Oh, another one that often happens is filtering secondaries. So secondaries is where you have your little gene fragment and you have 2 SNPs on that same fragment. And it's often just base standard to get rid of them, right? So, and that is the right thing to do if you are doing an analysis that is affected by recombination. So I'm going to presume that everyone here remembers their first-year biology, because I am a first-year biology lecturer, so I know you should know this. So recombination is when you have the crossing over in meiosis that splits apart genes that are alleles that are on the same chromosome, okay? So you're getting this mixing. And when we do things like structure type analysis to look at population structure, when we do phylogenetics, we presume that all of our different loci are independently inherited. And therefore, you're going to give, if you have a whole lot of secondaries, they're going to tell the exact same story. But that's not because they're two independent markers. They're actually just really, really, really close together on the genome. And so we remove secondaries where our analysis requires independent inheritance, and we expect that to be shown in the analysis. But it can often get deleted when we don't necessarily want to do this. So we have the alternative situation where we have two parental genomes coming together to create something like a heterozygous site. And my thought is so that's very different from recombination. We're actually just counting the number of sites in the genome where the individual has more than one allele. And that has nothing to do with independent inheritance, or little to do with independent inheritance. There's arguments. If it's within a population, that's still real, genuine diversity that the population has. And I would argue that you probably don't want to remove secondaries where the diversity is a result of fertilization instead of recombination. Yeah? Does that kind of make sense? So you often see this in scripts, and then I removed secondaries. And you're like, well, why? Yeah? So if you get me as a reviewer, I'll always be like, why did you apply that filter? And what we're going to do in the code is to actually look at where some of these things make a difference. What is the right filtering order? The answer there is not the right filtering order, because everything you do needs to be based on the principles of the analysis you want to use your filtered data on. I, as a starting point, do something where I generally create myself, and we're going to do this in a minute, a couple minutes. Where does the get rid of all the crap? First off, let's just create a whole file where I've got rid of the loci that just did not work well, and where I've got rid of the individuals that were really bad. And I call that in this file the GL clean file. So I first go through and create a file where I've kind of dealt with all my issues. Because you can't actually get to the point you have a data set you feel confident in for your analysis if you're also still trying to figure out which individuals didn't work, which loci didn't work. So your first step is to go and do some kind of cleaning. And then for a population structure analysis, I would do another thing here that I actually forgot to put in this script, but then I would filter for that population genetic assessment. So I would first filter much more strongly on call rate, knowing that population structure analyses don't like missing data. And then I would filter on minor allele frequency. You could do 0.05. My baseline, if I'm working on very small numbers of individuals, which I'm doing with some of the really threatened species, is to actually use this code, which is just removing singletons. Yes? Yeah.
Just to precise to you, small details. The new version of the filter math function on that diverse will actually take an integer as minimum allele count.
Sorry?
The new version of the function in the package will take either a proportional number, so a number below 1, or an integer that gets interpreted as a minimum allele count.
Yeah. So yes, so if you have very few individuals, so like my sunset frogs with my 66 individuals, for which I think 50 are going to work, I would just remove singletons for population structure analysis to make that run faster, because they just add computation time. Because it's a structure analysis, I would remove my secondaries because they require independent inheritance, so we need loci that are not similar to close to each other. And I would then do a read depth filter that I forgot to put in here, which was a lot more hardcore than I did. I just forgot to put it in. And then I would do my really expensive thing. Now I have the cleanest SNPs I can get. I've got rid of everything I can get rid of. And at that point, I'm going to filter my individuals. So remember the first time I threw away the really bad individuals. And at this step, I'm going to look at whether I have any other individuals who are not performing. And again, this is iterative. You're going to be doing this, and then you're going to run it and be like, why is this individual being weird? So you're going to go back and rerun your call rate filter and go, actually, that one has a lot lower, it works a lot less well than everyone else. So that's probably a sequencing issue. So you're going to be doing this multiple times when you try and run things. So what's right for one situation is probably wrong for a different one. So the important things are to be flexible, like let your data inform you of what's going on. You do need to really understand what each filter is doing and understand whether that filter is right for the question you are going to ask with the data set you output. And know that you are going to need multiple different sets of filters to ask different questions. So I think we could probably go to the code. I wrote this, I'm not very good at running code and talk, so I thought I'd talk, we'd run a little bit of code, and then I'll talk some more and we'll run a little bit more code. So if you open that up, I forget what line it is, so because of changes, we've swapped me to first, so does anyone know what line? Can I? Yes. All right. Does anyone have questions? Yes, microphone. Does someone want to run around with a microphone? There it is.
Just a quick question about reproducibility. With our samples, we normally run a lot of replicates. We use scat samples, so the quality is generally pretty poor. So does the filtering in that case merge the two samples, or does it just give like an indicator of how good it is?
So in the first run, it's used as an indicator of how good a locus is. If there's various ways to deal with it that have their own outcomes, like it is possible if you're running from raw data to merge your FASTQ files. It actually is super, super easy to do that, but then you have very different coverage for your individuals. So when I don't know exactly what Dart-R does with the reproducibility data or the reproduced individuals in terms of SNP calling. Andrew, are you able to answer that? So when you guys do duplicates for checking the reproducibility or the replicability, what happens? Do you merge all that raw data? No. So they return you a single individual. Ask them exactly what they do. Sorry, Mike.
Yeah, in a target file where all individuals are separated and then the final SNP scores, I think they're combined. There's some situations where I don't think it is. I have to come up to particular examples.
Okay. Rob, yes.
I can ask a really ignorant question, which is, other than you mentioned you filtered low frequency minor alleles to speed up the computation, what other reasons would you ever want to filter out data like that?
If you have really low read depth, the vast majority of your singletons will probably be sequencing error. And so you might not have confidence in them. So I'm going to talk about how important read depth is for particular types of metric.
To some extent, though, if you're also filtering on your read depth, doesn't that eliminate the problem?
That's the point, yeah. Yeah, so if you filter, well, it depends on your read depth you're left with. So if you have quite low read depth across the board, can't rely on your singletons very well, essentially. Yes.
Yeah, thanks. I was wondering how useful data is like with importing data. So if you have already generated like a variant calling file, like a VCF file,
Yeah.
and import it into a data
Yes.
as a generate object,
Yeah.
will you still be able to run all those filters and things?
So yeah, so all you do is ask for the code. So I told Rob I was going to say this. So the code I'm currently using is code that was given me by Kim Ottewell from DVCA in WA, who got it from Robin, and Robin has a whole chunks of it written by code from people at University of Sydney and the Threatened Species Initiative. And so just ask, because I think like a lot of us are also running from VCF files. There's a bunch of steps you have to do. Robin wrote some phenomenal code that she is happy to share. That actually lets you, that goes through and calculates all of those additional columns of data so that Dart-R works properly. So you don't have to invent the wheel, you just have to ask. And it is very doable.
Thank you.
I'll just throw in 2 comments to things that I would do in addition to what you've done. Yeah. So one is I always look at individual heterozygosity, because often we find in our data sets you'll get individuals with stupidly high heterozygosity that's clearly contamination or some error or something like that, and they always do funky things in your data set. It gets hard to judge how much heterozygosity is too much, but it's something you need to look at as well. And then what I usually do is once I've worked out what individuals I don't want, when I re-import the data, I eliminate those individuals from the very beginning and then reapply all my filtering, so that way you're not knocking off loci that might be on the borderline. If you get rid of the individuals with crap data to start with, then you improve your calls.
No, that's a good point. So let's say you've gone through this process and you think you know what filters you want to do on your data set, then yes, you would want to, I mean, ideally import a data set that SNP's called on just those individuals. In some scenarios, it doesn't matter if you just import it, remove those individuals, and then run the rest of the pipeline. Those are both options you can do, yeah. So we're just gonna have a quick go at running code for just running a structure type analysis. So if you go into your Gandalf, I thought because of Luis's age that this was gonna be all wizard because of Harry Potter, but I'm clearly wrong. So essentially,
yeah, maybe so if you go here in the RStudio Cloud so you have here in the working directory, and then there is one folder for each session. So we are in session 2. You click on session 2, and then this is this file, the QMD file, which is the Quattro format. Mm-hmm. Yes.
Okay, so we're going to do this a little bit out of order. So on this code, you're going to run, essentially load the packages. So we're going to run lines 17 through 38. However you need to do that. I don't know how to working.
Yeah, so to work here, each one of these squares is called a chunk. So if you want to run a chunk, you have just to play here, and it runs. And then you go to the next one, and so on.
Okay.
The play button.
Okay. So after you've done that, we're actually, so over here there's this outline. This is very helpful. So we're actually going to go down to these assessing population words that you can't see. Assessing population structure and demography. So this is just a quick pipeline. So this is Euparlia crassa. I'm a frog biologist at heart. So this is just two species in the Kimberley and Pilbara. Our section has disconnected, so I don't know what to do about that. But what you can do, so this is just an example to work through the set of filters. So it creates this GL clean file, and then does each of those filters in order, and then does a quick maximum likelihood version of structure called SNMF. And we'll just output a quick structure plot for the species. So it's just a basic, if I was going to do a quick, fast rule of thumb structure analysis, just to see what's happening, this is what I would do. Now it's working. So that's from line 183. Hate you. We have a long relationship. It goes back and forth, Rob.
Wait, does it mean we have to run everything about 1?
No, so you're just running those first little bits that were line. So just the 2 little chunks at the beginning that are loading all the libraries. Then on the side thing, go to assessing population. It's like star, star, assessing population structure. And we're just doing that section. So raise your hand if you need help. There's like 12 people here who are here to help, so if you're stuck. So this is just getting your head around some of the basic functions. Yes, Robin. Oh, yes. Thought you needed help. I was going to make Robin help you.
I just had a question. You run gl.filter monomorphs as its own line. I've always just added into the removing individual call rate, the mono remove.
Yeah, I've done that in later code, but I want you guys to recognize that that's an important filler. So GL filter monomorphs, after you remove individuals, what happens is, let's say you have a ton of those singletons, those single copy alleles in your data set. So every individual is going to have a whole bunch of those. If you remove that individual, you have loci that are now called monomorphic, because they're actually not SNP. There's no SNPs anymore. There's only, what's less than biallelic, monoallelic. And so that filter actually goes over each of your loci and says, oh, you removed this individual. I'm removing all the loci that are no longer variable. So yes, you can do that in the actual call rate filter, but I wanted to make a point that that's pretty important. Some things will just crash on you, like structure, if you don't do it. We're almost there. Okay. Right. All right. Run chunk. Okay, so we're running this first chunk to load first set of packages and this second chunk, and then I'm going to outline to this assessing, and I'm going to load in the data I gave you guys from this paper, which it's thinking about. So, I don't know, is it just me, or does run chunk run a lot slower than running the lines?
And the other thing is that you can do here, this is actually you can go here, and yes, it's the maximum velocity. Yeah, should work now.
So you can't make it faster. So I don't know, I've played with this, and sometimes I just find it faster to go run that line. Look, it did something. Yay. Okay, done. All right. So this is just a map to show you, Control Enter, okay. So this is just a map to show you where the samples are from this paper. It's very beautiful, Kimberly, top end, tiny little brown frogs. And then this is the first round. So this is cutting through and getting rid of your loci that worked pretty badly. And so this is, so you can look at each of them. I wish it would show up in plots instead of up here, but. So as you run all of this, I guess you just run the chunk. So you can see. So as you can see on the side, it's creating all of these steps. As it steps through each of them, every time I run a filter, it's creating a whole new thing, a new object. And it's going to produce GL clean. So what I have done is, so I looked at this. So I first looked at the call rate by locus, and I cut everything that was below 80%, so it had to be present in 80% of the individuals. And then so that's after I did call rate by individual. So you can see there was, after I cut those, there was a couple of individuals I got rid of, I think, because they weren't very good. And I went through this whole process, and I get to this final step, which is showing, this is what my sort of end individual matrix looks like. And this is cleaned up. This is not at all what I would use to run an analysis, but I've just got rid of everything that's kind of a mess. So I'm left with something. I feel confident that I can properly understand the filtering parameters. Then after that, I'm going to filter this for population structure analyses. One thing I didn't put in here that I would suggest is that you put in a call rate filter, because I forgot to put it in when I was typing this in the middle of the night. And so in this, what you can see, I've now put my call rate threshold to much, much higher. I've made it 0.98. And then I'm actually filtering out all my singletons. I'm getting rid of my singletons because we don't want those for our structure analysis. And then again, I'm checking my individuals and getting rid of anyone who's still bad. In this case, it actually doesn't remove anyone. And I've called this GL structure. This is the structure type analysis that I'm happy to do, and it leaves me with 36 individuals and just about 9,500 loci, which is more than enough for a population structure analysis. And that's what it looks like. So we can see, it's actually really easy to see our two populations. You can see the bands of red versus purple, so you're seeing those two populations come out, but you're also seeing that there's really not much missing data in this data set. So your answer here is going to be relatively obvious that it's two populations. So I've given you a little output code for a yes? Probably. What am I doing? Does anyone have questions on that? So this is sort of the steps for a structure analysis. Yes. Microphone. There it is.
Is there a reason why you apply the secondaries filter in the second step instead of at the very beginning, since it's a structure analysis?
I mean,
does it matter if you...
So why did I get rid of secondary SNPs later rather than earlier?
Because I normally do it basically at the very start.
So depending on, because the secondaries is basically often just randomly choosing one. So if you run the remove secondaries early, you might have a bad one that's going to get deleted and a good one you're going to keep, but you don't get to choose that. But by running it later, you're getting rid of all the bad ones, and if you still have secondaries, they're both good. So you're kind of artificially losing loci that you probably don't need to.
Yeah.
So I do secondaries much later so I can get rid of bad things before I get rid of secondaries.
Yeah. I know you can filter them depending on some, not randomly, like the polymorphism, information content. And, but probably, thank you.
Okay, so yes, we end up with our cleaned up data set. And so this is a maximum likelihood version of structure called SNMF.
Sorry, Renee?
Yes, another one. Yeah.
Sorry, I just want to mention that in Dart you can select the method for filtering out the secondaries.
Yeah.
So it has method and you select random, or you can select best. And
I don't really know what best means, so I just make sure that what's
Okay.
left is best and then select random. Let's- I would still do it later. I would still do it later so that because even if you use best, maybe the best one would drop out after some particular filter. So it doesn't make sense to me to remove them before you've got rid of bad things. If they're both good, great, then remove one. But get rid of bad things before you do that. Okay, so this is, I put a bunch of warning up above this. This is not the right way to do this for a publication. SNMF is great, it can be fast. Robin has great code for SNMF, but running it properly takes a lot of iterations, and running a bunch of tests of parameters called alpha intolerance, but this is gonna be fine for what we're doing. And when we've run that, we then get a plot that tells us our cross-entropy criterion. So I'm not going to go into the methodologies because somebody else is teaching you population structure. But the short answer is the value that's lowest is your best population structure. But 3 populations isn't bad. So we're going to decide on 2. And this produces a bar plot for us somewhere. What is happening? Okay, there we go. That's not what I wanted. I wanted this. So I get my nice 2 populations, which are pretty clear, and I had one very admixed individual. And when I just look at a quick PCA, which I like for exploring, but I never publish anymore because they're so upsetting in a huge number of ways. And so what we see again is our 2 populations, and then that center individual is actually just geographically central, because frogs in the north are pretty hard to get because you need a helicopter. So what you're actually seeing is not a lack of admixture, what you're seeing is a lack of sampling. So that's sort of a baseline structure filtering. Does everyone kind of make sense why we choose, feeling okay with why we chose the filters we chose? Okay. So I'm going to take us to here. Okay, and so, thank you, I appreciate that. So let's talk a little bit about other types of filtering. So you need to filter, how am I doing on time? Okay. So one of the key things we often try and do is delimit conservation units. And conservation units have been around since the 90s, and it's idea of how do we split like a threatened species into groups for management. And so this is bet hedging on the term species, but this black box is a species under current taxonomy. And then we have what are 2 ESUs. And those are 2 groups within the species that do not really have any gene flow between them. But within each ESU, we have sets of populations that do have gene flow, but that, as shown by the arrows, varies between them. And this is important information in conservation because what are the discrete units? Where is their gene flow? These are all really relevant. Like if a population gets extirpated or is undergoing demographic decline, like if we fix the declining problem, are they going to recover on their own? Are they going to get migration? There's lots of really important information that we get from this. And we have to be really careful about how we do this. So this is a paper I did, and the question we often ask is there gene flow? Now on this thing, what you can see is some really cool stuff, all right? So we've got the purple and the blue. So the blue here, this big blue blotch, is East Coast Australia, and this purple is Northern Australia, so Top End and Kimberley. This is Broome. This is Alice Springs. This is New Caledonia, and this is Tahiti, right? So for the conservation mind, I think this is great. These are discrete units, and I'm using this as a specific example about how structure tells us about gene flow, but it doesn't tell us about biology because this data set is for something else. This data set is for, where did it go? An invasive pest species, okay? So that first, if you just took a structure analysis and said, these are our management units and this is the history and therefore that's the management, you would be very, very, very wrong. Because you established that there wasn't gene flow, but you didn't establish why there wasn't gene flow. So you know it's not happening, but you don't know why it's not happening. So this is a tree mix analysis, another analysis where singletons are very important. And in this, it's a maximum likelihood phylogenetic tree of all our populations, and the branch lengths are genetic drift, which occurs for a bunch of reasons. In this particular case, founder effects. So what we've had, and this is supported by our knowledge of what's happened, is this native Australian East Coast fly that is here and destroys all of our fruit, appears to have had what actually appears to be a natural movement into the north, but then it's jumped into Broome. And Broome is actually quite inbred and drifted. But we also had it jump across the South Pacific into New Caledonia and fruit shipped there. And then from New Caledonia, it went to Tahiti. And so we've had repeated founder effects. So I use this as a cautionary example of overinterpreting. You know, structure-like analyses tell you there's no gene flow, but they don't tell you why. And it's the same for lots of things. Fixed difference analyses are a little bit of a pet peeve of mine in conservation because people interpret them as management units. So there are fixed differences. The maths behind fixed differences is totally valid, but there are different ways fixed differences accrue besides them being distinct evolutionary units. So detecting fixed differences says there's a lack of gene flow. And often I read papers and I say, oh, it's a new subspecies ESU, we should manage it independently, and the answer is maybe. We have to go back again to our first year genetics. Hardy-Weinberg equilibrium. Genetic variation in a population will remain constant from one generation to the next in the absence of disturbing factors, okay? So we expect all of these things gene flow to occur unless there's mutations, natural selection, non-random mating, genetic drift due to population size or gene flow from other areas, from other populations. Two of these are really, really important in conservation. Non-random mating and genetic drift due to population size. You can accrue fixed differences because you have become isolated through anthropogenic change. So it's really important not to jump from there is structure to this is a distinct and unique population. If you have a really highly structured species in Hardy-Weinberg equilibrium, probably they're different. So this is a paper I've just got accepted on Latoria rubella. It is hyperabundant. I just did some field work looking for it, peed in the toilet, flushed the toilet, 5 came out. That's for real. That was 3 weeks ago. Conservation, you don't get to flush a toilet and have 5 of your target species come out. So this is hyperabundant, but what you can see is a complete lack of gene flow across the East Coast, right? So in this species, there is nothing that suggests this is an issue where There's non-random mating, genetic drift, right? So we're not having inbreeding, we're not having genetic drift, which means that gene flow is actually stopped, and that means we have different species, okay? So this is the fundamental underlying premise of all population genetics when we're looking across landscapes. So in this case, concluded they're different species. Found one sounds like this, ah, and the other one sounds like this, ah. That's frogs. Okay. But if they're not in Hardy-Weinberg equilibrium, you need to be careful because you could end up having the misidentification of inbred and genetically drifted populations identified as discrete conservation units. And that's by thinking that analyses that show you structure also illustrate the causal factor behind what's creating that structure. So you have to be very careful. This is a great paper. I've put a link into it. So this is by Kate Rick, a PhD student at UWA, led by Kim Ottewell, who's a fantastic conservation geneticist for DBCA. And it's on the golden bandicoot. And this is one of those species that this blue here was the distribution of the species before European occupation of Australia. And now it lives on these islands here, the Northwest Kimberley and Barrow Island in the Pilbara. And in this paper, essentially, although you could get all sorts of differences, and they actually showed basically a complete lack of gene flow between each of those populations, they did not conclude not to mix them. They actually concluded, particularly the islands in northeast top end, desperately need supplementation from the Kimberley, which is the most diverse population. And so it's really important to have that biological and historical context, and you need to ask, is there gene flow? But then you also need to clearly evaluate, why is there not gene flow? And so this is another species I've worked on a bit. So this is the rufous hair wallaby. And it's a really, really hard one because it now lives mostly here. It used to live with these red dots, but went extinct, except they took the last 20 and put them in a captive breeding population. And it also used to be here. And when you have a species like this, where you're trying to understand what's happening, you have to remember things like Bergmann's rule, Allen's rule, island rule. Things get bigger. Small mammals get bigger on islands, and big ones get smaller. There's aridity gradients. There are latitudinal gradients and things like that. What that also implies is there was allele frequency differences across this range. So you can get fixed differences and various other things because they had allele frequency distances and probably isolation by distance, but you actually just cut out the middle, right? So the allele frequencies here and here were different. And if you cut out the center where there was admixture, you just end up with fixed differences. So you have to be really, really careful not to end up with negative effects. So we just did this bit. So I wanted to come back to singletons. I did this a bit, I'll go quickly. So again, a single allele in one individual in a data set. And these are really vital to things like heterozygosity, so the presence of two alleles at a gene locus in an individual. Tegemas D, so Tegemas D is a really good metric to kind of look at contraction and expansion. And it compares the observed nucleotide diversity against the expected diversity, presuming a constant population size and neutral and constant population size. And we're gonna actually play with that one in this next session. So does tree mix, so genetic drift involves like rare alleles going to high frequency through founder events. So if you get rid of your rare alleles, you're going to mess up your estimations. And relatedness and inbreeding are also impacted somewhat by this type of filtering. So the issue we have to deal with in these kind of questions our read error. And again, this is our confidence in our alleles. So this is stacks, what stacks does. Let's say this is all the sequences for one gene across a population. And what it's taking is creating each allele as a stack of identical loci, and then it takes those stacks and go, actually, these two are the same gene. And that's how you get your heterozygosity. Each of those are identical, but when you put them on top of each other, you see the differences in a gene between individuals. And to get that confidence, you need preferentially a higher read depth. And that's really important. So to deal with singletons and ask those questions, you need a read depth where you're confident. And so I just kind of modified the filtering for structure analyses from that clean data set. And so oh, I forgot to change the top line, but we still filter for call rate, but we have got rid of, we had to do a strong read depth filter, very strong read depth filter, because we want to be confident in these alleles. Sorry? Yes. And then we remove our monomorphs. So we're not getting rid of secondaries, we're not doing minor allele frequency, but we're pressing our read depth filters up higher, so we're very confident in those really rare things. And I just want to say before we do this last little bit that not all data sets can answer every question, so you need to be thinking about this, and I'll talk tomorrow about planning. Even if you can run the analysis and get an output, you need to make sure that your sampling and your sequencing answer the questions you want to answer. If you're looking at population structure, you don't necessarily need really high read depth, but if you want to answer really hardcore population genetic questions, you need higher read depth to be able to keep those rare alleles. If we go to cloud, we're on to this next bit. And so I'm gonna let you guys run through this on your own, but this is just showing you what happens to this Chijima's D metric if you filter in different ways. And then thinking about what the outcome is of actually doing it incorrectly. So this first bit is a function that I wrote to calculate Chijima's D. So it just writes a function over there. And it filters first using minor allele frequency filtering, and then no minor allele filtering, and filtering with no minor alleles and on read depth. So if you guys want to run through that, and then I'll be done. This starts from exercise. Right on the side bit is exercise. Yes. Do we have a microphone?
How do you know what the right answer is when you said you filter it various ways and then see what's the right answer? But obviously, if you don't know going into it, it's difficult.
That's why I called this the art of filtering. The recommendation is you need to do it a whole bunch of different ways. You need to see what filters your answer is really sensitive to. So I've never published a paper where I haven't run the whole pipeline at least like 45 times. It's just what happens. So running it over and over, looking at sensitivity to particular filters. If it's insensitive to a filter, well, it doesn't matter. If it is sensitive to a filter, you need to think about, well, what is the right thing given the actual analysis and the math theory behind the analysis that I'm doing? And does this make biological sense? If it doesn't make biological sense, then you've probably done something wrong. So there's no way to know that this is absolutely the right answer until we can do whole genome sequencing at high depth on every individual for very cheap. And that will probably solve our problems a little bit more, but we're not there yet.
That's just a minor question, but you filter for the individuals very late. And I think you kind of throw away a little bit of loci by doing that as a last step. If you do it first, get rid of the really bad individuals, that could save you some of the loci.
This is running on the GL clean, so we already did that first filter step. So this here is running on, so remember early on we just did a get rid of our worst loci, get rid of our bad individuals. That's running on that data set.
I thought you did loci, call rate loci first, and then you do call rate loci individuals later?
Yes.
On the GL4? And I wonder why that is.
Because I want to narrow it down to the best loci I can get before I throw away my individuals.
Yeah, and as I say, and I would argue it's better to get the really bad individuals out there.
Yeah,
first,
no, I did that. So what you see,
do
this
the
is
other one.
running in the GL clean data set.
Oh, they're already there. Okay, good.
So we already did so what I had said earlier was before you start run with these sort of really lenient filters, which is something I stole from Robin. And so really lenient filters, and then I'm moving on from that data set where I've cleaned out all the crap first.
Okay.
Yeah.
Can I answer his question in a different way? So if you care more about having more loci, more individuals in your final data, you want to eliminate bad loci first. If you want more loci, eliminate bad individuals first. Does that make sense?
Yes.
Because if you're eliminating loci, you're making what individuals you leave behind better. If you eliminate individuals first, you make the loci you leave behind better.
Yes.
So it depends what your priority is, loci or individuals.
Well, I've been talking about expansion and structure, so
Right,
I
so
care
you'll-
more about individuals most of the time. And
Thank you. Renee, I've got a question, please. When you said earlier that before you published a paper, you would have run it through the thing 45 or so times.
yes.
Are you being flippant?
No.
Or, okay, so just to help me understand, after each of those steps in the 45, what are you looking for? Do you have a structure that you follow that says, okay, at this point
Yeah,
I'm
I'm
checking
just
that one
looking
again?
at, like, is that, what does my data set look like? What is its completeness? Who have I got rid of? You know, for a particular analysis, I would say I wouldn't run it 45 times, but across the paper. So every time I'm looking at, have I done the filtering well? What's happened to my individuals? Did something weird happen? Do I need to go back and reassess a particular individual? Maybe I lost too much, and maybe I can go and, you know, make a few filters less stringent, but is that going to actually change my answer? And so when you do that, you have to play with those and see what happens.
Okay, thanks for answering the question. It raises more, but thanks.
Yes, it does, yeah. Feel free to tell me if I'm wrong on the others, but I mean, I'm looking at various people, and I think most people are like, yeah, kind of, yeah.
On that, I think there was a question before about how do you keep track of what you've tried, and do you have some kind of log of what the results of the different
Learn how to
changes were?
structure your outputs really well. So I'll have like a code that I've run for something in particular, and I'll have like a separate folder for every output with different parameters. And I will, like other people have different answers, but I will just have, and so the way I tend to write code is I'll have a block where it specifies output names and things like that. So I can say, oh, this is what I did in the bottom, and it's going to output to this folder. And I can copy that code and just alter a couple things so I can go back through them. And then when I've come to the right ones, I delete huge amounts of stuff out of my Dropbox.
Hello. So tomorrow we have Yaya tomorrow that is going to exactly speaking about that, how we can reproduce our research and keeping track of everything. So don't miss it.
So sorry, I'm just running this last bit. So did everyone able to get through that code, or? Oof, okay. I'm not a fan of this. Well, we'll just look at these first two. Oh, you have to do view in this. Oh, there it is. With and that.
I'm just kicking quick in. There's a GL print history on the GenLight object, and it tells you actually what kind of filter you have done. So that that gives a little bit of track. I'm not sure. We haven't pronounced that very often. I don't think many people use it, but I think it's useful.
Yeah. So what I wanted to do is just show you the difference here. So with Tajima's D value, a D value that is positive is an indication of populations undergoing contraction, and a positive value is a measure of a population undergoing, or a negative value is expansion. And so where we actually cut out all of those singletons and did a standard 0.05 minor allele frequency, we can see both of our populations are positive, and one is quite positive to the point it probably would be significantly positive, which says that both populations are contracting. Seems rather unlikely. These are, again, I work on garbage frogs. They are everywhere. Possibly in the Northern Territory, the most abundant frog in the Northern Territory, undergoing what could be significant contraction. That seems quite unlikely. But when I look at that look at this without removing those minor alleles, keeping those singletons, and being confident in those singletons, which is really important because that's actually the maths that so implies the Tajima's D calculation, what I see is a fundamental change. And when I did the statistics on this, that you actually have to do a simulation to get it, a p-value, we got a significant expansion process in the Kimberley and a non-significant expansion process in the top end. And this actually made a lot of biological sense and went along with all of the other data. So I wanted to do this as an example of where you could get, where you're filtering, just following basic steps of filtering that seem really common, could lead you to actually the totally incorrect answer. And so it's really important that your filtering steps match the theoretical expectations of the metric that you are going to actually calculate. And that's where I'm at for today. So we have questions. Do I get a beer? Did you say beer? Oh.
I had just one thing to add, which I mentioned to Bernd earlier, that one shortcut that I sometimes use for analyses is to do correlations while I'm filtering. So, you know, you have your quality metrics or your quality filters like call rate, read depth, repeatability, average, and I guess none of those should be correlated with any kind of biological metric. So sometimes I use that to gauge how much to filter. If, I don't know, FIS is correlated really strongly with call rate, maybe you're losing heterozygotes, and then bump that up until you don't have a correlation.
Yeah, I would say maybe we can get your code up in where everyone can get to it, because it not only does all of that, but it also brings in the VCF file from Stacks and calculates the metrics, and it's wonderful, and I've stolen it. Yes.
So I would throw in an opposition to you. So we mostly do phylogeography, structure-y type things. We've normally got fairly dense sampling, but low individuals per population. It almost doesn't matter what we do for the filtering, our answers don't change. So it's not always that you have to be super thorough with every aspect of your filtering. As you said, totally depends on your question and your approach.
Yeah, I mean, honestly, spatial analyses with good sampling are the ones that are going to be the least sensitive to your filtering. But where you're doing demographic analyses, you can really, really change your answers. Yeah. So Tajima's D, yeah, if you use the package, sorry, the question is, can you calculate Tajima's D on any type of data set? I'm sure you can. I mean, he wrote it, Tajima wrote it when it was like very old. But so the function I gave you is written for SNPs because there's a part of it that is a ratio, so you have to know how many individuals it actually worked on for it to work. So the herfstat and popgen are package functions for Tajima's D, don't deal with missing data as if it were sequence data, and so they delete columns. And I think you guys have you done Tajima's D yet as a SNP thing? I've given you the code like 6 times. You have it now. So that function is written particularly for SNP data, and I had Matt Hahn, who literally wrote a book called Population Genetics, which I totally recommend. I get no money, but it's a fantastic guide. Matt Hahn's book, it's H-A-H-N. Matt Hahn's book is a great primer for understanding like what the maths are behind all of the different things. And so I asked him to check that code, so I feel confident that it's right. And you have it now. Yeah.
So I was wondering what your experience has been with unequal population sampling, like subpopulation sampling.
Depends on the question.
If you're looking for like trying to delimit species, for instance.
So this one, in that paper was really interesting. It's not in the paper, but if you had really unequal sample sizes and you called SNPs on them, particularly with an outgroup present, it did weird things where that paper's presented, it was just the 2 species looking at detecting hybrids, and it's almost 50-50. And there was slightly unequal sample sizes, but if you have really unequal sample sizes and you add a bunch of noise by having other groups, what I found is you can get really weird things, like the initial runs where I used SNPs called without groups, I was getting like Q values, and that's the proportion assigned to one or the other population of like 0.65. And it was biased towards the population that was most well sampled. Right. So my F1 hybrids were somehow more of the population had greater sampling from. Okay. And so I think they can be a little bit sensitive, but I don't think population structure analyses are like the least sensitive of all things, I think, that you can do.
Sorry, can I have one more question? Luis, can I have one last question? Why do you hate PCOAs? Why do you hate PCOAs?
Because people think they mean something other than a measure of some splitting variation. Like one interesting thing you can do is if you run a bunch of populations with an outgroup, your outgroup ends up like dead center in your PCOA. So it's just trying to partition the variance, and it's like somewhat meaningless. And it's interesting, every time, if you call SNPs with outgroups, it affects them in interesting ways. So let's say you have a species with an outgroup, and you include that closest species in outgroup, what happens is it makes your PC1 between your target populations higher and PC2 less, and the more outgroups you add, the more it shifts your variance to PC1. And then people say things like, it's 42% variance, so it's different species or management units, and you're like, no, that's not what it means. There's like a lot of interpretation. So I think I hate them because people give them far too much credence in what they actually mean. I think they're great as a tool to start to visualize what's coming out of your data, but I basically try not to even publish them now.